Our goal today is to talk about how does meiosis create cells that are different from each other. Another way to say this is how does it create variation? We know that every single person on this earth is unique and they all have different DNA for the most part. And so how does that happen? Well, we know that each sperm and egg that are created in meiosis are genetically unique, meaning they are different. But how does that happen in meiosis? Well, there's a few things that happen in meiosis that don't actually happen in mitosis, and that's what we're going to talk about. The first thing, right, is that each person has 46 chromosomes. They have a pair, one from mom, one from dad, one from mom, one from dad. And this is in every single one of their cells. These are called homologous chromosomes. So homologous chromosomes, they're similar in size and shape. They also have similar traits, so eye color and freckles. They might not have identical traits because this one codes for blue eyes, this one for brown eyes, but they both code for eye color. And the reason is because maybe this is from dad. Dad gave a brown-eyed gene and mom gave a blue-eyed gene. These are not homologous because they're different sizes and shape and they don't code for the same thing. So we're going to write down this definition for homologous chromosomes because a lot of the stuff that happens to make our sperm and egg cells different have to do with these homologous chromosomes. So the definition here, chromosomes with the same types of traits that are similar but not identical. Feel free to pause this so you can continue writing it down, but I'm going to move ahead. So the first way that we can get different sperm and egg cells every single time we go through meiosis is through something called independent assortment. If we break down this word, an assortment means lots of different ones. So we have lots of different chromosomes, and we're going to make lots of different types of reproductive cells, lots of different sperm, lots of different eggs. Independent means the chromosomes can move independently. They're not stuck to each other. They don't have to go in a specific order. They're independent. In mitosis, if I have these chromosomes line up, in this case, we're just using two. I've got a red and a blue, and I switch them to be a blue on top and then a red. We're still going to get the same cells. They both still have a blue and a red chromatid in them. So it doesn't matter how my chromosomes line up. In meiosis, they line up a little differently. Notice how they're in pairs here. How they line up determines what combinations of chromosomes are in these gametes, in these reproductive cells. So here I've got blue on one side, red on one side, and I end up with blue and then red chromosomes in the cells. Here I have them switched, I have them mixed, and we end up with mixed chromosomes in the cells. So this is the idea of independent assortment, the idea that chromosomes can line up in different ways, and they'll produce different daughter cells depending on how they line up. This happens in metaphase, so when they're lining up in the middle. All right, moving on. If you want to draw a picture in your notes here, might be a good idea. If you don't have colors, don't worry. You can just make one like a really dark colored chromosome and make one a light colored chromosome and just show them lining up in one way and then in another way. We also have crossing over. So this funny thing, these homologous chromosomes, when they line up in pairs with their homologous pair, they actually trade parts of their chromosome. So parts of this chromosome is going to get cut off and switch with this other chromosome. And so we end up with chromosomes with different combinations. So go ahead and write this down. Homologous chromosomes swap parts during metaphase. Lastly, we have something called non-disjunction. Notice here we have my chromosomes, and two of these chromosomes are supposed to be going to separate sides of the cell, but they're going to the same side. So non-disjunction is when chromosomes don't go to the correct sides of the cell. They don't split, and then we end up with extra chromosomes in one cell and fewer in another. It can happen in a variety of ways. This is in the first metaphase. This is what happens. We end up with 3 and 1 instead of 2 and 2. If it happens in the second one, then we've got a 3 and 1 here, and these cells will be normal. So it depends on if it happens in the first metaphase when they line up or the second one. But either way, it's when chromosomes fail to separate during meiosis. Most of the time, the sperm and egg that have extra chromosomes or fewer chromosomes don't end up surviving. On some rare occasions, we do have some disorders. So some individuals can survive and be born, and there are some disorders, one of the most common ones being Down syndrome.